Let's now talk a little bit about microbial evolution and the diversity that exists of microbial life on Earth today. There are three main domains recognized currently by the biological community. Specifically, this is held within the field of taxonomy, which is the science of classification of living things. All life on Earth, this of course would exclude viruses and prions, are classified into one of three domains: the bacteria, archaea, and eukarya. The first two of these, the archaea and bacteria, more specifically known as eubacteria and archaebacteria, are of the cell type known as prokaryotic cells. This means they are lacking the nucleus and instead have DNA found in a nucleoid, do not have organelles, and are generally considered small. They are also all unicellular organisms in these two domains. These were the first cell types found on Earth, roughly 4 billion years ago. Your book initially says that life arose 3.5 billion years ago, but then later on in the chapter talks about fossil evidence indicating life existed up to 3.8 billion years ago. We in fact have current fossil evidence indicating life might have existed even up to 4.1 billion years ago. And so for that reason, we can sort of round off to roughly 4 billion years of life on Earth. Earth itself was formed 4.5 billion years ago. So what this indicates is that really quickly after Earth formed and started to cool, life evolved on Earth. These cell types, the bacteria and archaea, the prokaryotic cells, were the only cells on Earth for about 2 billion years. Then, roughly 1.8 billion years ago, the eukaryotic cells evolved. This was a brand new domain, and from this domain comes cells with a nucleus that have organelles, are generally larger, and have the capacity to form multicellular organisms. This three-domain classification system was proposed by Carl Woese in 1990 and is widely accepted by biologists today. However, classification of living things is constantly changing, and we may see classification of living things change again within your lifetime. But this is our best guess as to how we can classify all life on Earth today. This is based on their genetics, their biochemistry or molecules found inside the cells, and other known characteristics. From this, we can learn a little bit more about the three different domains of life. In Table 5.3 of your book, it goes over the three different domains and talks very briefly about different characteristics found in each. Now, if we talk about the evolution of life on Earth, the universal common ancestor of all life on Earth would have lived about 4 billion years ago. This common ancestor is now known today to have evolved in deep sea hydrothermal vents, essentially underwater volcanoes. Those organisms were most like some of the archaea that are alive today. This is one of the reasons why archaea are named archaea, because they are archaic or ancient. This ancient form of life that formed deep in the oceans near these hydrothermal vents or underwater volcanoes is where life first is thought to have evolved. From that archaeal domain, bacteria would have also evolved, roughly 3.5 to 4 billion years ago. These two domains then branched off and formed separate living structures and evolved on their own. And then, as I mentioned, about 2 billion years ago, the eukarya evolved. Eukarya evolved out of the archaeal domain with some contributions from bacteria, as we'll learn about later. But the eukarya are thought to have also branched out of archaea. So archaea is most like our universal common ancestor 4 billion years ago. As mentioned, the first life on Earth arose in these underwater hydrothermal vents. Those hydrothermal vents provided heat energy and was also likely the source of chemical energy for these chemosynthetic autotrophs that lived deep in the oceans. Then, roughly 3.5 to 3.8 billion years ago, bacteria had evolved that was able to live on the surface of the ocean and not deep in the ocean. Living on the surface of the ocean provided these bacteria with the available resource and endless energy of sunlight. These bacteria are still alive today, and we refer to them as cyanobacteria. Cyanobacteria are small unicellular organisms that are photosynthetic. They tend to color the waters green, much like unicellular algae do. The green color of the water does not necessarily indicate that it is eukaryotic algae. In fact, these cyanobacteria are photosynthetic prokaryotic cells, so much smaller, don't contain chloroplasts, and don't contain a nucleus. But they all look the same to the human eye. It's only under the microscope that you can notice the difference. 3.5 to 3.8 billion years ago, these cyanobacteria were photosynthesizing, grabbing energy from sunlight and turning it into chemical energy like sugars and fats. As a result of their activity, they produced mucus secretions that eventually solidified and formed stromatolites, which look like rocks to us, but in fact are comprised of cemented organic materials from so long ago. Importantly, these cyanobacteria were not the first life on Earth, because the first life on Earth was thought to have evolved much earlier and deep in the oceans. But these cyanobacteria contributed significantly to life on Earth and the chemistry of our planet. When life first arose 4 billion years ago, the Earth's atmosphere was mostly carbon dioxide and nitrogen, and almost no oxygen at all. There were a few other inorganic gases, but no organic gases existed. It wasn't until these microorganisms, these cyanobacteria, about 3.5 to 3.8 billion years ago, started releasing oxygen into the atmosphere as a poisonous byproduct of photosynthesis, that our atmosphere eventually changed. Today, oxygen accounts for approximately 20% of our atmosphere. During the time of dinosaurs, it was in fact over 30%. So oxygen concentrations have fluctuated over geologic time. However, the biggest difference was when the Earth's atmosphere went to essentially no oxygen gas to 20 or 30% oxygen gas, all as a result of the growth of these cyanobacteria for hundreds of millions of years. For hundreds of millions of years, they were spewing oxygen gas into the atmosphere, and this oxygen was being released because it was toxic. If they held on to it, in fact, they would die. So instead, they released it into the atmosphere where it accumulated and turned eventually into ozone. As sunlight and UV radiation hits oxygen gas in the atmosphere, those oxygen molecules become ozone molecules. And ozone molecules can accumulate in the atmosphere as well, eventually, after hundreds of millions of years, forming the ozone layer. The initial concentration of oxygen in the atmosphere eventually killed most of life on Earth and was in fact probably the cause of our first major mass extinction, where just as life was getting started on Earth, it was almost all killed off by these cyanobacteria. But thankfully, a few living systems were able to evolve the ability to live in oxygen and detoxify this poisonous gas. Out of those organisms, even more evolution occurred to the point where some microorganisms were able to not only detoxify oxygen, but actually use it in their metabolism. Those organisms are now our aerobic organisms. And today, they comprise most of life on Earth, because most of life on Earth is exposed to oxygen gas. So these little guys were significant contributors to life on Earth and our entire planet. There are lots of microorganisms that live all over our planet and inside living systems. There's in fact a huge variety of microbial diversity on our Earth today. These cyanobacteria still exist, but so do many others. One of these is a group of microorganisms known as extremophiles. Extremophiles live in environments that we consider extreme because we could not live there. Either they're too hot, too cold, too acidic, too salty, something. But organisms, microorganisms, have evolved the ability to live there. This doesn't necessarily make them hardy. They, in fact, may eventually die in our environment because our environment might be too cold for them or too hot for them. What this means is that they evolved to live optimally in their own specific environment. However, because they live in such an extreme environment compared to our lifestyle, we refer to them as extremophiles. There are lots of different types of extremophiles. One type are the thermophiles, of which there's also the hyperthermophiles. Thermophiles and hyperthermophiles live in really hot environments, at or near the boiling point of water. You can find these in Yellowstone National Park, or even closer to home here in California, in Lassen National Park. In these hot springs, the geothermal hot springs that often have beautiful colors to them, and the colors and pigments look a lot like different colors as a result of microbial growth. The different pigments are how some of these thermophiles and hyperthermophiles absorb solar energy. One of these thermophiles, Thermophilus aquaticus, was isolated from hot springs in Yellowstone and has become an essential role player in molecular biology labs around the world, because from it, an important enzyme was harvested, Taq DNA polymerase, which is used for a chemical reaction known as polymerase chain reaction, or PCR, that we'll be talking about later. There's also psychrophiles, which grow in really cold environments, like snow and ice. Halophiles, like we see here, that grow in really salty environments, like salt evaporation ponds. But the ocean is not salty enough for them. Remember, they're extremophiles. There's even methanogens that produce methane and live in the stomachs of cows or in swamps and contribute to the methane gas produced in these locations. In the next section, we'll talk about the different types of microbes that we're going to study this semester, because we won't be focusing on these extremophiles very much longer.